hypothalamus releasing and inhibitory hormones are secreted into the median eminence. Special neurons in the hypothalamus synthesize and secrete hypothalamic releasing and inhibitory hormones that control secretion of the anterior pituitary hormones. These neurons originate in various parts of the hypothalamus and send their nerve fibers to the median eminence and tuber cinereum, an extension of hypothalamic tissue into the pituitary stalk. The endings of these fibers are different from most endings in the central nervous system in that their function is not to transmit signals from one neuron to another, but rather to secrete the hypothalamic releasing and inhibitory hormones into the tissue fluids. These hormones are immediately absorbed into the hypothalamic hypophyseal portal system and carried directly into the sinuses of the anterior pituitary gland. Thyrotropin releasing hormone peptide of 3 amino acid and stimulates secretion of thyroid stimulating hormone by thyrotropes. Gonadotropin releasing hormone single chain of 10 amino acids stimulates secretion of FSH and LH by gonadotropes. Corticotropin releasing hormone single chain of 41 amino acid stimulates secretion of ACTH by corticotropes. Growth hormone releasing hormone single chain of 44 amino acid stimulates secretion of growth hormone by somatotropes. Growth hormone inhibitory hormone single chain of 14 amino acid inhibits secretion of growth hormone by somatotropes. Prolactin inhibiting hormone dopamine catecholamine inhibits synthesis and secretion of prolactin by lactotropes. Hypothalamic releasing and inhibitory hormones control anterior pituitary secretion. The hypothalamic releasing and inhibitory hormones control anterior pituitary secretion. The function of the releasing and inhibitory hormone is to control secretion of the anterior pituitary hormones. For most of the anterior pituitary hormones, it is the releasing hormone that are important. But for prolactin, a hypothalamic inhibitory hormone probably exerts more control. The major hypothalamic releasing and inhibitory hormones, which are summarized in the table, are the following: thyrotropin releasing hormone, which causes the release of thyroid stimulating hormone; corticotropin releasing hormone, which causes the release of ACTH; growth hormone releasing hormone, which causes the release of growth hormone, and growth hormone inhibitory hormone, also called somatostatin, which inhibits the release of growth hormone; gonadotropin releasing hormone, which causes the release of two gonadotropes hormone, LH and FSH; prolactin inhibiting inhibitory hormone, also known as dopamine, which causes inhibition of prolactin secretion. Additional hypothalamic hormones include one that additional hypothalamic hormones include one that stimulates prolactin secretion and perhaps other that inhibit release of the anterior pituitary hormones. Each of the more important hypothalamic hormone is discussed in detail at specific hormonal system controlled by them are presented in this and subsequent chapters. Specific areas in hypothalamus control secretion of specific hypothalamic releasing and inhibitory hormones. All or most of the hypothalamic hormones are secreted at nerve endings in the median eminence before being transported to the anterior pituitary gland. Electrical stimulation of this region excites these nerve endings and therefore cause release of essentially all hypothalamic releasing hormones. However, the neuronal cell bodies that give rise to these median eminence nerve endings are located in the discrete areas of the hypothalamus or in closely related areas of the basal brain. Physiological functions of growth hormone. All the major anterior pituitary hormones except for growth hormone exert their principal effects mainly by stimulating target glands, including thyroid gland, adrenal cortex, ovaries, testicles, and mammary glands. The function of each of the function of each of the function of each of these pituitary hormones are so uh, intimately concerned with the function of the respective target glands that except for growth hormone, Their function are discussed in subsequent chapters along the target cells. Growth hormone exert its effect directly on or almost on all or almost all tissues of the body. Growth hormone promotes growth of many body tissues. Growth hormone, also called somatotrophic or somatotropin, is a small protein molecule that contains 191 amino acid in a single chain and has a molecular weight of 22,005. It causes growth of almost all tissues of the body that are capable of growing. It promotes increased sizes of the cells and increased mitosis, with the development of greater number of cells and specific differentiation of certain types of cells such as bone growth and early muscle cells. Table 76.5 shows typical weight uh, charts of two growth hormone, two growing litter mate rats. One received daily injections of growth hormone, another did not receive growth hormone. This figure shows marked in enhancement of growth hormone in the rat given growth hormone in the early days of life and even after the two rats reach adulthood. In the early in the early stages of development, all organs of the treated rat increase proportionality in size, and after adulthood was reached, most of the bones stopped lengthening. But many of soft tissues continue to grow. Mm, uh, once the epiphyses of the long bones have united with once the epiphyses of the long bones have united with the shafts, further lengthening of the bone cannot occur. Even though many other tissues of the body can continue to grow throughout life. Growth hormone has several metabolic effects. Aside from its general effect in causing growth hormone has multiple specific metabolic effects. 76 figure 76.6 including the following: increased rate of protein synthesis in most cells of the body, increased mobilization of fatty acid from adipose tissue, increased fatty acid in the blood, and increased use of fatty acid for energy, decreased rate of glucose utilization throughout the body. Thus, in effect, growth hormone enhances body protein and decreases fat store and conserve carbohydrate. Growth hormone promotes protein deposition in tissues. Although the precise mechanism by which growth hormone increases protein deposition are not fully understood, a series of different effects are known, all of which could lead to enhanced protein deposition. Enhancement of amino acid transport through the cell membrane. Growth hormone directly enhances transport of amino acid through cell membrane to the interior of the cells. This increases amino acid concentration in the cells and is presumed to be at least partially responsible for the increased protein synthesis. This control of amino acid transport is similar to the effect of insulin in controlling the glucose transport through the membrane. Enhancement of RNA translation to cause protein synthesis by ribosome. Even when the amino acid concentrations are not increased in the cells, growth hormone still increases RNA translation and causing protein to be synthesized in greater amounts by the ribosome in the cytoplasm. Increased nuclear transcription of DNA to form RNA. Over more prolonged periods, 24 to 28, 48 hours, growth hormone also stimulates transcription of DNA in the nucleus, causing formation of increased quantities of RNA. This promotes This promotes protein synthesis and growth if sufficient energy, amino acids, vitamins, and other requisites for growth are available. In the long run, this may be most important function of growth hormone. Decreased catabolism of protein and amino acid. In addition to increase in protein synthesis, growth hormone increases breakdown of cell protein. A probable reason for this decrease is that growth hormone also mobilizes large quantities of free fatty acid from the adipose tissue, and these are used to supply most of the energy for the body cells, thus acting as a potent protein sparer. Summary: growth hormone enhances all facets of amino acid uptake and protein synthesis by cells, while at the same time reducing the breakdown of proteins. Mm, Growth hormone enhances fat utilization of energy. Growth hormone has a specific effect to really cause release of fatty acid from the adipose tissue and therefore increases the concentration of fatty acid in the body fluid. Also in tissues throughout the body, growth hormone enhances conversion of fatty acid to acetyl coenzyme and its subsequent utilization for energy. Therefore, under the influence of growth hormone, fat is used for energy in preference to use of carbohydrates and proteins. The ability of growth hormone to promote fat utilization together with its protein mm, the ability of growth hormone to promote fat utilization together with its protein anabolic effect causes an increase in lean body mass. However, mobilization of fat by growth hormone requires several hours to occur, whereas enhancement of protein synthesis can begin in minutes under the influence of growth hormone. Ketogenic effect of excess growth hormone. Under the influence of excessive amounts of growth hormone, fat mobilization from adipose tissues sometimes mm, becomes so great that large quantities of acetoacetic acid are formed by the liver and released into the body fluids, thus causing ketosis. This excessive mobilization of fat from adipose tissue also frequently causes a fatty layer. Growth hormone growth hormone growth hormone decreases carbohydrate utilization. Growth hormone has multiple effects that influence carbohydrate metabolism, including glucose uptake in tissues such as skeletal muscle and fat, increased glucose production by the liver, and increased insulin secretion. Each of these changes results from growth hormone induced insulin resistance, which attenuates insulin's reaction to stimulate uptake and utilization of glucose in skeletal muscle and adipose tissue and to inhibit gluconeogenesis, glucose production by the liver. This leads to increased blood glucose concentration and a compensatory increase in insulin secretion. For these reasons, growth hormones effects are called diabetogenic, and excessive secretion of growth hormone can produce metabolic disturbances similar to those found in patients with type 2 non-insulin dependent diabetes who are also resistant to metabolic effects of insulin. However, patients with acromegaly who have excessive growth hormone secretion are usually lean with little visceral fat, whereas patients with type 2 diabetes are frequently overweight with excessive visceral fat, which drives insulin resistance. We don't we do not know the precise mechanisms by which growth hormone causes insulin resistance and decreased glucose utilization by cells. However, increases lipolysis and blood concentrations of fatty acids uh, likely contribute to impairment of insulin action to on tissue glucose utilization. Experimental studies indicate that raising blood levels of fatty acid above normal rapidly decreases the sensitivity of the liver and skeletal muscle to insulin's effects on carbohydrate metabolism. Necessity of insulin and carbohydrate for growth promoting action of growth hormone. Growth hormone fails to cause growth in animals that lack a pancreas. It also fails to cause growth if carbohydrates are excluded from diet. Thus, adequate insulin activity and adequate availability of carbohydrates are necessary for growth hormone to be effective. Part of this requirement for carbohydrate and insulin is to provide the energy needed for metabolism of growth, but there seem to be other effects as well. Especially important is the ability of insulin to enhance the transport of some amino acid into cells in the same way that it stimulates glucose transport. Growth hormone stimulates cartilage and bone growth. Also, Although growth hormone stimulates increased deposition of proteins and increased growth in almost all tissues of the body, its most obvious effect is to increase growth of skeletal frame. This results from multiple effects of growth hormone on bone, including increased deposition of protein by chondrocytic and osteogenic cells that cause bone growth, increased rate of reproduction of these cells, and three, a specific effect of converting chondrocytes into osteogenic cells, thus causing deposition of new bone. There are two principal mechanisms of bone growth. First, in response to growth hormone stimulation, the long bones grow in length at the epiphyseal cartilages, where the epiphyses at the ends of bone are separated from the shaft. This growth first causes deposition of new cartilage, followed by its conversion into new bone, thus elongating the shaft and pushing the epiphyses further and further apart. At the same time, the epiphyseal cartilage is progressively used up, so by late adolescence, no additional epiphyseal cartilage remains to provide for further long bone growth. At this time, at this time, bony fusion occurs between the shaft and the epiphysis at each end, so no further lengthening of the long bone can occur. Second, osteoblasts in bone periosteum and in some bone cavities deposit new bone on the surface of the older bone. Simultaneously, osteoclasts in bone discussed in chapter 8 and detail in chapter 80 remove old bone. When the rate of deposition is greater than the resorption, the thickness of the bone increases. Growth hormone strongly stimulates osteoblasts, and therefore the bones can continue to become thicker throughout life under the influence of growth hormone. This is especially true for membranous bones. For example, the jaw bones can be stimulated to grow even after adolescence, causing forward protrusion of the chin and lower teeth. Likewise, the bones of the skull can grow in thickness and give rise to bony protrusions over the eyes. Growth hormone exerts much of its effect through insulin like growth factors, somatomedians. Growth hormone causes the liver and to a much lesser extent other tissues to form several small proteins called insulin like growth factors, IGFs, and also called somatomedians that mediate some of the growth and metabolic effects of growth hormone. At least four IGFs have been isolated, but by far the most important of these is IGF-1 somatomedian C. The molecular weight of IGF-1 is about 7500 and its concentration in the plasma closely follows the rate of growth hormone secretion. Children with deficiency of IGF fail to grow normally even though they may have normal or elevated secretion of growth hormone. The the pygmy people of Africa, for example, have very small stature due to congenital ability to synthesize significant amount of IGF-1. Although their plasma concentration of growth hormone is either normal or high, they have diminished amount of IGF-1 in the plasma, which apparently accounts for the small stature of these people. Some other dwarfs, Laren syndrome, have a similar problem, generally caused by mutation of growth hormone and therefore failure of growth hormone to stimulate formation of IGF-1. It has been postulated that most of the growth effects of growth hormone result from IGF-1 and other IGFs rather than from the direct effects of GH on the bones and other peripheral tissues. Even so, experiments have demonstrated that injection of growth hormone directly into the epiphyseal cartilages of the bones of living animal causes growth of these cartilage areas, and the amount of growth hormone required for this growth to occur is minute. One possible explanation of this finding is that growth hormone can cause formation of enough IGF-1 in the local tissue to cause local growth. However, growth hormone also has IGF independent effects that stimulate growth in some tissues such as chondrocytes of cartilage. Short duration of action of growth hormone but prolonged action of IGF-1. Growth hormone attaches only weakly to the plasma proteins in the blood. Therefore, it is released from the blood into the tissues rapidly, having a half time of blood of less than 20 minutes. By contrast, IGF-1 attaches strongly to the carrier protein in the blood that, like IGF-1, is produced in response to growth hormone. As a result, IGF-1 is released only slowly from the blood to the tissues with a half time of about 20 hours. This slow time release greatly prolongs the growth promoting effects of the breast of the growth hormone growth secretion. Regulation of growth hormone secretion. After adolescence, growth hormone secretion decreases slowly with aging, finally falling to about 25% of the adolescent level in very old age. Growth hormone is secreted in a pulsatile pattern, increasing and decreasing. The precise mechanism that control the secretion of growth hormone are not fully understood, but several factors are related to a person's state of nutrition or status are known to stimulate secretion. Starvation, especially with severe protein deficiency, hypoglycemia, or lower concentration of fatty acids in the blood, exercise and excitement, trauma, ghrelin, a hormone secreted by the stomach before meals. Some amino acids, including arginine, growth hormone, also characteristically increases during the first 2 hours of deep sleep. Figures 76.3 summarizes some of the factors that are known to influence growth hormone secretion. Normal concentration of growth hormone in the plasma of an adult is about 1.6 to 3 nanogram per milliliter in a child or adolescent is about 6 nanogram per milliliter. These values may increase to as high as 50 nanogram per milliliter after depletion of the body stores of the protein or carbohydrates during prolonged starvation. Under acute conditions, hypoglycemia is a far more potent stimulator of growth hormone that is an acute decrease in protein intake. Conversely, in chronic conditions, growth secretion seem to correlate more with the degree of cellular protein depletion than with the degree of glucose insufficiency. For example, the extremely high levels of growth hormone that occur during the starvation are closely related to the amount of protein depletion. Figure 76.8 demonstrate the effect of the protein deficiency on plasma growth hormone and then the effect of adding protein into the diet. The first column shows the first column shows very high levels of growth hormone in children with extreme protein deficiency during the protein malnutrition and condition called kwashiorkor. And the second column shows the levels in some children after 3 days of treatment with more than adequate quantities of carbohydrates in their diets, demonstrating that the carbohydrate did not lower the plasma growth hormone concentration. The third and the fourth column shows the levels after the treatment with the supplements for 3 and 25 days respectively, with a concomitant decrease in the hormone. These results demonstrate that under severe conditions of protein malnutrition, adequate calories alone are not sufficient to correct the excess production of growth hormone. The protein deficiency must also be corrected before growth hormone will return to the normal.